Welcome back to the course. In the last class we were looking at 2D NMR spectroscopy. We looked briefly at how the data is acquired, how the magnetization is transferred from one nucleus to another nucleus. So, we will continue with that today. So, this is the slide which was shown last time, which showed the schematic of how a 2D NMR experiment works. So, as I said, we start from applying first an RF pulse to the nucleus, any given nucleus. So, let us say this is one hydrogen proton that excites this particular proton. So, it brings from z axis to xy plane. By now you must be now familiar with the concept of going from z axis to xy plane when we apply a 90-degree pulse. So, you have to basically recollect that part or recall that part, that when you apply a 90-degree RF pulse, you excite the magnetization which goes from z axis to xy axis or x or y axis. Now, once that happens, we remove the RF pulse and the spin simply rotates or precesses in the xy plane. During this process, it is also recovering to the z axis and also it is dephasing in the x and y plane. So, this is the relaxation part which is going on here. During that process, we transfer the magnetization from A to B. So, that is what we do through some kind of a interaction. That means, two hydrogens, two protons A and B should have an interaction between them, some kind of an interaction, and typically there are two types of interaction in NMR which we use for transferring from one spin to another. One is called as J coupling through bond, and other is dipolar coupling through space. So, both of this we will use in this course, we will see in this course how it can be used. For now, let us assume that there is only J coupling between A and B. So, these are two hydrogens in a given molecule, they are, let us say, separated by 3 bonds or even 2 bonds. So, there could be a geminal coupling or a vicinal coupling. So, during this evolution, so we use the word evolution is the time when the spin A is free to rotate in the xy plane or precess in the xy plane. So, this is during that time the spin A precesses with the frequency of A, because that is the precessional frequency, and that time period is labeled as T1. This is one time period. So, this is an FID. So, it should be, I will see in the later slides, we will write it as cosine omega A T1. So, now this during this period when it is evolving or when it is precessing, at the same time simultaneously transfer from A to B is happens. Now, how does that happen? As I said, that is through the mechanism of J coupling in this case. So, this is called mixing. You are actually basically transferring, but you are not excited the B pulse. So, it is not mixing in that sense, it is essentially just transferring. So, once the transfer has happened, we then apply another RF pulse like similar to this, which acts on both A and B. So, it is not an RF pulse which works only on A, it will also act on A and B. So, that is that mixing pulse, it mixes the magnetization which has come from A and now that gets transferred to B. So, now the B nucleus gets excited similar to what happened here, and therefore, that spin B now starts rotating or precessing after this pulse mixing pulse is applied or after the second RF pulse is applied, it starts precessing with frequency of its own, which is omega B. Now, one thing which I mentioned in the last class is this transfer is not 100%. Usually, it is a very small fraction of magnetization from A which is transferred to B. Majority of the magnetization of A remains on A itself, it is not transferred to B. Even though the J coupling may be strong enough, the coupling in proton to proton, in case of proton to proton, it is not so efficient. So, therefore, your magnetization remains a bulk of it, majority of it remains on A. So, there are two magnetizations now, when you finally detect, both of them are detected, and these are independent, they are different, they are separated, but they come together. So, now when I do a Fourier transform, I will see a pattern like this. So, here is plotted a T1 axis here and T2. Actually, when you do Fourier transform, we end up with frequencies. So, this is just T1, T2 is just a schematic to show what happens during that period, but actually what you will see in a real spectrum is this frequency scale. So, you can see here for spin A omega A, it was during T1 omega A and this combination of omega A and T2. So, if you look at these two combination, you can see that they are both omega A's. So, that happens here, you can see this is this particular peak. here it has omega A during the T1 in the vertical axis, and it is also having omega A during T2, which is the horizontal axis. So, therefore, that is a peak which has just no particular information, it just has a correlation of A with itself. So, we do not have any useful information in this peak, but the other combination where you go from A to B. So, you see here if I take this combination where it is evolving or it is frequency evolution is omega A during T1, it then evolves with omega B during T2. This combination basically will tell us that omega A is during T1, that is this T1 axis coordinate, and during T2 it is omega B, if I draw a vertical line here. So, this peak is now a very useful peak for us, because it is telling us that there is an interaction or correlation or connection between two spins A and B. So, this information would not have been possible to obtain in from 1D NMR. 1D NMR would have given A peak separately, B peak separately, but no such cross peak. So, we use the term cross peak in 2D NMR. A cross peak is basically a peak between two different spins which are correlated or connected or interacting via J coupling or through dipolar coupling. So, now this is as far as A spin is concerned, but remember I mentioned in this just before some time that your spins, the RF pulse is applicable to both A and B. So, which means if I can excite A in the beginning here, that should also excite B, the same RF pulse. The same RF pulse here and here should work on both A and B. So, now the B gets excited also at the same time as A. So, this and this are happening simultaneously. So, B evolves similar to what happened to A here, and B has a frequency omega B during T1. Again the same process happens, if B is coupled, A is coupled to B, B also will be coupled to A. So, this mixing or transfer of magnetization is the same process as here, but only thing is it is A to B here and here it is B to A. So, it is a reverse. Now, after the transfer has happened, we apply another RF pulse, that is a mixing pulse, which will now excite the one which was not transferred. So, for example, here B was excited, it remains excited, it further goes away. I mean this is the magnetization which remains on B, that as I said majority of it remains on the nucleus, it does not get transferred, whereas the one which got transferred to A gets excited, the A nucleus is excited by the second RF pulse, and that then evolves during the detection time. This is where we physically detect the signal. Here this evolution period there is no detection. Okay. So, this is something one should keep in mind, that we have two T1 values, two time values T1 and T2, but the actual detection, the physical, the detector hardware detects signal only during this period T2. This is indirect detection, this is not actually detected, but then because we capture this combination in a spectrum, we can call this as a indirect dimension. So, this combination now let us see how it comes in the spectrum. It will be similar to this, except it is now coming at the B frequency. So, you can see during T1 this frequency which is B and this is also B. So, this peak is similar to this here A to A, and why this is B to B, this is this combination omega B T2 with omega B T1. So, therefore, this peak has no useful information, it is not telling me to what it is connected or correlated, whereas this peak, the second one here, this tells me that omega B and omega A are connected. Now, this is similar to this peak, here also the information was omega A is connected to B. So, you see we get the same information two times. So, now these two have to be combined together, because as I said both these processes are happening simultaneously. So, this is not that this happens separately from here. So, when both are together, the spectrum also will be a combination of these two. So, when you combine the two spectra or two patterns, essentially what you get is a spectrum like this. So, this is the 2D NMR spectrum. So, you can see here omega A to omega A and B to B, and that lies neatly on a what is called a diagonal peak of a square. So, this is like a square. Why is it a square? Because you have the same frequencies here, the same scale and the same frequency and scale here. So, now these two peaks are the cross peaks. They are the useful ones because they give us the information that A and B spins are correlated. So, you can see that we are getting actually redundant or information twice. So, in principle you may say why do we need two times, but that is the design of the experiment is like this, and sometimes it helps because a peak here will be overlapped with some other peak, whereas its partner which is exactly the same may not be overlapped. So, if you cannot identify the cross peak here, you may be able to identify the cross peak here or vice versa. So, it is useful to have duplicates twice the same frequency, same core information in case of overlaps. So, this is what now is shown here, is that you call this dimension, that is the indirect dimension, that is the T1 axis. We are not detecting the signal physically here. There is an FID here, because anything evolves in NMR by a frequency, that is an FID, but that FID is not captured or digitized or detected by the hardware or RF coil. It modulates the intensity of the direct dimension. This is the dimension where actually we see detect physically the signal. The signal here, whatever is coming out here, is detected and Fourier transform, and similarly it is modulated by this here. So, we Fourier transform here as well, because here also we have an FID, but it is not an induction decay, in the sense it is not inducing a signal in the coil directly, it is only indirectly affecting the signal here. So, these are the basic concepts one should keep in mind when analyzing or understanding 2D NMR, that is diagonal peak and cross peak. As we go later, a simple one slight complication will come, is that in a heteronuclear NMR, in there we will see that later, but you can in heteronuclear NMR what will happen is this axis will be a heteronuclear, means carbon or nitrogen, whereas this axis will be proton. So, therefore, they will not be a same axis and scale. So, you will not get a diagonal peak in heteronuclear, because you will never detect carbon carbon or proton proton correlation when you are recording a carbon proton experiment. So, we will see that point later, but it is important to keep in mind this diagonal peak business or cross peak concept arises mainly in homonuclear NMR, where both axes are either proton and proton or both are carbon and carbon. So, let us now go little bit into mathematical detail of how a 2D NMR experiment is made, because unless we go a little bit into mathematics of this, it will be difficult to capture or gather information on 3D when we go little bit more complicated experiments. So, let me little bit give a little more detail insight now. So, this period before this pulse is applied. So, this is the first RF pulse. If you recall the previous slide, we have said we apply RF pulse on spin I. This pulse actually is the pulse before which we have to prepare the system in equilibrium, and that is similar to 1D pulse sequence I showed you in the previous class when we did 1D part, and there we had this called as relaxation delay, and that depends on the T1 of your sample. So, similarly here this preparation period depends on the T1 relaxation value of the sample or the molecule. Then you apply this RF pulse, and during this period is what we saw, it is evolution, and once you evolve there is this omega into T term will come, and during second RF period, during this period from here to here, that is during evolution, the magnetization is actually transferred from spin 1 to spin 2, spin A to spin B, and that results in another pulse which will cause it to excite the transferred nuclei, and that will now evolve with its respective frequency. So, that is the detection part which I showed you in the previous slide. So, this is a schematic of a 2D NMR experiment. So, you start with a preparation period, you have an evolution period, you have a mixing period, and then again you have an evolution period. So, one has to keep in mind that preparation period is not just relaxation delay, you are also preparing the magnetization to ready to be transferred or evolve. So, therefore, it also can consist of those initial pulses, RF pulses, the 90 degree which we apply. During the evolution period, one has to keep in mind there is no pulse applied. So, during this evolution period, typically we are letting the spin free to just simply evolve and relax. So, there is a relaxation also happening in this time, but normally we do not consider relaxation when we analyze the correlations. So, we are looking at 2D NMR spectral correlations. Relaxation is not so important to be worried about or concerned about. So, we assume that we are not allowing it to relax fully, we are not allowing to go up to the end. Typically the evolution part is stopped here somewhere after it reaches about 20 to 30% of its signal, and then the second pulse, the new set of pulse, that is the mixing pulse, is applied. So, mixing pulse is basically now doing the job of transferring actually the polarization or exciting the second set of nucleus from where to which the magnetization has been transferred. And the final detection is again a simple FID which decays with time, and this is allowed to complete. We do not stop this in between, we allow this to complete fully. So, your question may arise in your mind that why is that here we stop somewhere in between, but why is it that we do not do that here. And this is a very important point in NMR. This is related to what is called as a measurement time. So, what happens is, suppose let us say I have here 100 points, or let us say this whole set of FIDs, if I digitize, meaning if I take collect points, let us say it corresponds to 1000 points, and let us say this also corresponds to 1000 points. So, what happens is, for every point here I will have to record 1000 points here. So, how the experiment is conducted is basically you record one point, you evolve this magnetization during T1 for a single point, and then after that point you apply the mixing pulse, then you detect the full signal. Then you apply, then you go back to the preparation period here, wait for the equilibration, apply the pulse, then go for a second point. That means you evolve it for 2 points. That initially was point 0, now it is 0 and 1, and then again you apply the mixing pulse, so and apply take this full. So, if you have 1000 points here and 1000 points here, here is 1000 points will take probably 100 milliseconds, but for every point here you have to record the entire FID here. So, therefore, if I have to do each point here, so let us say the whole cycle, this whole cycle, let us say it takes about 2 to 3 seconds, then if I have to record 1000 points, I have to wait for 1000 into 3 seconds, because each point I do here will take the entire cycle 5 seconds. So, I have to come back, increase this point, go back, record full, again come back, and so on. So, the measurement time goes up very fast, because in 1000 times 5 seconds or 3 seconds I have to record this if I have to record the full FID. Therefore, we normally cut it down, we cut it down to about 200 or 300, let us say here 1/3 of it, and that saves the time, because now I have to do 200 times 5 seconds or 3 seconds, depending on how long is this entire sequence. So, therefore, an indirect dimension in NMR 2D, 3D and 4D. In fact, in 3D and 4D you will have 2 more dimensions like this in 3D. So, there we cut it even more. So, you can see as we go to higher dimensions, the signal time becomes very large, because of this constraint that every point you acquire here you have to repeat the whole cycle again increment it with another point and repeat again the entire cycle. You can't finish this fully and immediately go to this, you have to do this step by step, but this can be done continuously. So, this is the technical problem, and therefore, it is cut down, and then some other techniques are used to recover this whatever is the cut down signal. So, the resolution basically is affected by this problem. So, here since remember in NMR, if you allow the signal to completely go to 0 and if you get this full FID, your resolution is very good, you get the best resolution. But here, because I am now truncating, so we use this word called truncation. So, if I am truncating the signal to only a few 1/10 or 1/20 or 1/3 of the points, my signal resolution also gets down by 1/3. So, the line becomes broader. So, that is the drawback in multidimensional NMR spectroscopy, that we get broad signals, and that broad signals is coming because we have the limitation of not recording the data fully. We can only record data as to 1/3 or 1/4 depending on the time. So, there are varieties. One of the research of area of research in NMR is how to actually improve the resolution even if I am not completely getting the data. So, let us say I stop here at 20%. So, remaining 80% is not recorded. So, can I still do something to improve my resolution, because my resolution will be very much badly affected if I do not record this full FID. So, this is area of research, we will not go into detail in this particular course. So, this is the schematic of a 2D NMR. This is a relaxation delay, RF pulse, these are again set of RF pulse and so on. So, this is what I said, we have to repeat this n number of times, and that depends on how many scans. So, this is, so there are 2 delays here, one delay in the sense there are 2 constraints here. One is the relaxation delay, which is of the order of seconds, and the other constraint is how many times you have to repeat the experiment to get a good signal to noise. So, we use the word scans. So, more the scans, better is the signal to noise, but more the scans, longer is the experiment going to take. So, this essentially your sensitivity very much depends on these factors, how much time you record the data, how much is the number of scans and so on. So, these are the set of NMR experiments 2D which we will go through this in this course. We will not be able to go through all of them though. This for example, COSY toxy is something which we will see. Inadequate is something not normally done in biological samples. So, we will not go through that was actually covered in the previous NMR course. And nosy and rosy are very important for us because the entire structure determination of proteins depends on this experiment nosy, and rosy is not so much done for proteins, it is mainly for peptides, but we will still go through it and get the feel of how these experiments work. We will also look at this is a homonuclear experiment wherein both the axes are hydrogen and hydrogen, and whereas in this case we will see heteronuclear experiments where one of the axis is hydrogen, the other axis is carbon or nitrogen. So, let us start from the very first experiment, this is 2D correlation spectroscopy. This is one of the very first NMR experiment also which was developed and slowly it evolved into many more variants. So, the current experiment which we 2D COSY which we use is no longer the one which was originally published. There were subsequently many other published versions, but we will go to the most earliest version because we just want to get an idea of how this works and this experiment actually the principles of it. So, this COSY experiment is the simplest 2D NMR experiment you can think of. It essentially is the same similar to what we just discussed about the 2D NMR. So, you start from applying a 90-degree pulse which can be along x axis or it can be along y axis. You can assume it is x axis, then you apply this pulse. Once you apply your spin A which is along z axis, because before this pulse this is in equilibrium during this preparation period, it now comes into the xy plane or x axis. In this case because x pulse is applied it should be y axis, but does not matter right now. Let us assume that it is now in the x or the y axis. Now, during this period T1 it precesses. So, you see this arrow is shown as anti-clockwise, you can think of it as clockwise or anti-clockwise. It precesses or goes around the z axis. So, z is now vertical. So, you have to please keep in mind that this is a vertical axis. So, x and y plane is horizontal. So, this green circle which is shown here is actually is a plane, xy plane. So, now it tries to go in the xy plane and that precession mathematically is written like this cosine omega into T1. So, this is just simple rotation of the spins, the red color in the xy plane. Now, during that period when it is precessing, we transfer the magnetization to B through this J coupling concept, if there is a J coupling between A and B. So, that means the minimum requirement is that there should be a coupling between A and B, two spins. Now, once you do this transfer to B, now the B has to be excited because B is still along z axis. We are, although I said we excite both A and B here, but the magnetization which has come from A to B is still along z. B also will be xy plane, but again it transfers to A and the transferred part is along z. So, you have to keep in mind when I say that B has to be excited, it means it is the spin magnetization which has come from A, that part has to be excited. It is not the spin B has to be excited. In the beginning A and B both are excited by this pulse. Okay. So, this is something which you have to go back to the previous few slides ago, where we showed that A and B are equal and opposite. So, whatever happens to A happens to B, but the transferred portion from here to here, when we transfer the magnetization and the spin B part which is transferred is still along z, and that has to be brought into xy plane. That is done by applying another pulse, and this is what I said we call it as a mixing pulse. And once you do that, the spin B is on xy plane now, and A is also in the xy, because remember A does not transfer all of its magnetization to B, only part of it is transferred, the remaining part is still on A. So, A also remains like this, is rotating, B also. So, both of them are rotating now in this during this period T2. Okay. So, that is basically what is this mechanism. So, this is basically A and B both are correlated only because A and B both are connected to each other by J coupling. Now, if you look at it from the B point of view, you will have this concept, you will have the idea, you will have it a term called cosine omega B into T1, because A and B are equal and same, and during this T2 you will have cosine B into T2, cosine A into T2, omega A into T2. So, A and B will be reversed during T1 and T2, and this, so these two are the cross peaks which we saw in the previous slides. So, what is the advantage of this experiment homonuclear COSY? It correlates two proton chemical shifts or carbon chemical shifts that are directly coupled through J coupling. So, as I said, this J coupling is a very important requirement. Without this you can't transfer the coupling, transfer the magnetization from one spin to another. Diagonal spin corresponds to a 1D spectrum, and this is something which I mentioned last class also. The diagonal peaks actually in 2D NMR, homonuclear NMR, do not carry any information. They are actually a redundant information. They are only telling us A is connected to A, which anyway we know. So, a diagonal peak is actually a very difficult thing to get rid of. So, again a research problem in NMR has been to how to remove diagonal peak from a 2D homonuclear experiment, and because anyway that does not carry much information. And the problem with and second problem is it has a very strong nature, peak intensity. So, diagonal peak is something which comes with very high intensity and it spoils the spectrum several times, many a times. So, we like to normally get rid of it, but it is something not so easy to do that. So, we have to live with that. And coupled spin, how do we identify whether two spins A and B are coupled? They can be identified by simply looking at the cross peaks. So, the cross peak is the main important information carrying peak in the 2D spectrum. So, this is how a 2D COSY spectrum looks. This is very simple schematic, which is similar to what I showed you in the previous few slides ago, that you have a diagonal peak followed by you have a cross peak. So, these two basically are the most important for us, and again they are repeated, that actually duplicates whatever information is here is same as here. It's, as I said, it's good to have duplicates, because one peak may not give enough information because it may be crowded, you may not be able to extract the value, whereas the second peak may be useful, and vice versa. And whereas this diagonal peak is something which has no value to us, and therefore. So, this is basically what is shown here. You need basically coupling between A and B. So, here is shown as a vicinal coupling, meaning there is 1, 2 and 3 bond hydrogen to hydrogen, proton to proton coupling, or you can have a 2 bond coupling. But very rarely you can have a coupling between this proton and a very far away. So, COSY actually exploits up to 3 bond. 4 bond coupling and 5 bond is very difficult to detect with a COSY experiment, and therefore, we can't actually go beyond a few atoms, carbon atoms. So, if you want to do that, let us say you have a molecule which has a long chain which has, let us say this 4 5 carbons, and then if you want a connection or correlation between this hydrogen and an hydrogen located far away, we then have to use a TOCSY experiment, which we will see next. So, right now let us see what are the other things we can see with a COSY experiment, and this is the cross peak and diagonal peak which I already referred to previously. So, let us take a simple example of a molecule. So, this is this propyl chloride, a single chain amino acid, a molecule, a straight chain. So, you can see this is carbon number 1, carbon 2, carbon 3, and this is the 1D spectrum of the proton for proton 1, proton 2, proton 3. So, proton 1 is closest to chlorine, so it is most downfield shifted, proton 2 is slightly away, so the inductive effect is less, and proton 3 is farthest from the chlorine, so proton 3 is having the least inductive effect. So, this is 1D NMR spectrum. You can see that if I draw a diagonal peak now, so diagonal peak is simply correlation of 3 with 3, 2 proton 2 with 2, proton 1 with 1. So, that is nothing but the same 1D spectrum drawn in a diagonal way. Okay. But now let us see the cross peak. So, you see now here this hydrogen 1 and 2 are actually 3 bonds away. If you calculate, this is a vicinal hydrogens. So, if they are equivalent, I mean let us assume they are equivalent here, then these 2 protons are 3 bonds away, and therefore, in a COSY spectrum they will give cross peak, they are correlated, because 1 and 2 are close to each other by 3 bonds. Similarly, 2 and 3 are also connected to each other, because 2 and 3 are away by 3 bonds. So, they will also show a cross peak between 2 and 3. So, this is a pattern which you will expect in a COSY spectrum. You will not see any correlation between 1 and 3, because 1 and 3, these 2 hydrogens are really far away, they are 5 bonds separated. So, therefore, it is very difficult to or 4 bonds separated. It is very difficult to get information about them by simply doing a COSY. COSY will not work, because the coupling between 1 and 3, J coupling between proton 1 and 3 will be very small. So, this is why we will not see any peak here because of no direct coupling. So, this is an example of another real spectrum. This is taken from a book, Neil Jacobson's, and you can see here these are all different atoms. So, these are all different correlations observed between different atoms of the different hydrogens. So, you can see here only those two hydrogens protons which are directly coupled by J coupling are correlated in this spectrum. So, that means 7 will be correlated with 6, 6 will be correlated with 7 and 5, 5 with 6 and 5 with 4, 4 with 5, but 4 will not be coupled to 2 because 2 is far away. There is a carbonyl which is coming in between. Similarly, here 2 will be coupled to 3 and 3 will be coupled to 2, but within only these two you will not see any coupling the other spins. So, this brings us to the end of 2D cozy spectrum. In the next class we will look at 2D Toxi experiment, which helps us to actually go to the full chain here, except of course, there is a break here, but I will be able to see in a Toxi spectrum correlation from here this number 2 to 3 all the way, this number 2 to 7 or 4 to 7. So, that information will come from Toxi and we will look at it in the next class.